Hello, everyone, and welcome. Welcome, welcome. We have a number of folks coming in here. Um, Good morning, good afternoon, good evening, based on wherever you are. We're happy to have you with us for today's webinar, Understanding Clinical Trials with expert Dr. Andrew Beaton from AstraZeneca. And AstraZeneca is sponsoring this webinar. TMA is very grateful to AstraZeneca for their support as a corporate advisory council member and as a sponsor of our Global Myositis Patient Conference, MyoCon, and World Myositis Day. Today's webinar is the first in a series of 3 webinars that we hope will help the myositis community better understand the drug development process and help TMA better understand what kind of additional resources you all would like to consider clinical trial opportunities, to understand regulatory decision making, and to understand how pharmaceutical companies go about their important work to advance science and bring us closer to new therapies. So I'm really pleased to introduce today's special guest, Andrew Beaton from AstraZeneca, who has held senior leadership roles in end-to-end drug development, formerly with GSK, and now at AstraZeneca as the Global Clinical Program Leader, where he manages global clinical projects, which means that he defines clinical strategies for launch, provides expert opinions and advice in autoimmune disease, leads regulatory communications, and leads study physicians and scientists in the initiation, execution, completion, and data interpretation of clinical studies. There is truly no one better to draw back the curtain on drug development and clinical trials and to help all of us learn more about the history and future of new therapies, especially in myositis. So over to you, Dr. Beaton, with our gratitude for being with us today. Welcome.
Lovely. Thank you very much, Paula. And it really is my pleasure to be here with the Myositis Association to be able to start this webinar series. And I've got to say, obviously, this is very close to my heart, being a disease area that I'm currently working in at the moment. And it was a pleasure to meet you and the team in the US earlier this year. It was a great interaction and fantastic to meet some of the patients and hear their stories, because I think sometimes people feel that within industry, we do this drug development and that the patients are not necessarily at the forefront of our minds. They maybe think that we're big corporate entities looking at a financial well-being, but that couldn't be further from the truth. For us within AstraZeneca, patients are central to everything that we do. And for that reason, I'm so glad to be able again to meet with the TMA and provide today's webinar. I think for some have joined here maybe expecting something a little different, but I wanted to start from the absolute beginning for everyone, really, and understanding really how have we got to this current position and understanding clinical trials as they are. In future webinars, I will be talking about specific myositis clinical studies, but today I want to take people through a journey and understand why clinical trials are as they are today. So, how did we get here? What is the drug development process? What is our top level overview of clinical studies? And what does that mean to you as patients? What is your journey through those? And then I'll close with a couple of remarks. But I think moving forwards, we've got to think about, how did this all begin? Where was it that we really got to this point? So really, when we think about clinical trials, we need to think about, what are we trying to achieve? What is it that the patient needs? So clinical trials, in essence, test the safety and effectiveness of a drug, a therapy, a device. And to do that, we have to follow very strict processes that are there to protect study volunteers, but also to ensure that the data that we generate enables us to make decisions and be able to say that this is a meaningful product. But essentially, where we are now is very different to where this really started. And history really has shaped where we are with the conduct of modern clinical trials and the ethical frameworks. So where did it all begin? So pre-biblical times, 500 BC, we had King Nebuchadnezzar. Essentially, you could say back then, things weren't much different to today. There were always wars. There was always a desire to make sure that you as a nation were safe. And how better to do that, but to make sure that your people are always battle ready. So he thought, I've got this great idea. I think meat and wine will keep my soldiers battle ready. But a few vegetarians refused, and he allowed them to eat legumes, beans, and drink water for 10 days, thinking, no chance these are going to be as battle ready as my guys eating meat and drinking wine. Interestingly, at the end of the 10 days, those vegetarians were healthier than the meat eaters. And essentially, he allowed them to continue eating their vegetables moving forwards. Now, this wasn't a controlled study in any way, shape, or form, but at least he looked at two different groups and saw the differences between them. Moving on a little more, we can get to, the 16th century and a fortunate discovery made by Ambrose Parr. So he was a battlefield doctor. So back in those days, if you had injuries, the best remedy at the time was to pour boiling oil mixtures onto the wounds. So you essentially burnt and cauterized them, stopped them bleeding. Now, unfortunately, Ambrose ran out of boiling oil, so he had to think about alternative ways to treat his soldiers. He decided, for no reason other than he had it, to use egg yolk mixture instead. But what he found the next day is those that were treated with the egg yolk treatment actually had slept well, they were in less pain, they had no swelling. So obviously, although the boiling oil had been used for a long time, actually it was causing more damage than good. So this was just a lucky happenstance that he came to find an alternative way of treating those wounded on the battlefield. So moving on a little bit more, and we move on to James Lind. So he was a Royal Navy surgeon, and we know the stories from back in the day that, all the sailors, pirates even, they often had scurvy. Now, what was causing this? No one really knew. There were some people eating rats on ships that actually weren't suffering as badly. But he thought, I know what, I'm going to do a scientific experiment. I'm going to take 12 of these sailors and put 2 of each onto different treatments. So 2 of them, he put on a quart of cider every day. Probably quite nice if you enjoy your alcoholic drinks. 2 of them, he put onto elixir of vitriol, essentially a very dilute sulfuric acid. 2 he put onto half pint of seawater a day. 2 he put onto 2 oranges and 1 lemon a day. 2 onto vinegar 3 times a day, and then 2 he put onto this mixture of a paste of garlic, mustard seed, horseradish, and the likes. Now, what actually happened then as a result? Interestingly, those 2 sailors who were on 2 oranges and 1 lemon a day, they actually responded to the treatment. So their sores started healing. But the interesting thing was, although he'd made this great discovery, actually the Royal Navy never implemented this for a number of years later, having not really truly believed that this was what was going on. Now, if I go back to the rat eaters, now, interestingly, those sailors who were eating rats, unlike humans, can produce their own vitamin C. So those who are eating rats were actually getting the vitamin C from the 2 orange, comparable to the 2 oranges and 1 lemon a day, that are actually keeping them healthy. So again, some, by lucky happenstance, through their desire of rat flesh, were also self-medicating. So moving a little bit further forward now, you look at Austin Flint. So here we have an American here who was looking to treat hospital inmates suffering with rheumatic fever. But what he looked at that time was basically, could he give them, compared to a standard of care group who were getting treatments that were supposedly good for rheumatism at the time, he treated others with something called a placebo. So this is diluted tincture of quasar, which is essentially a bit of tree bark that has then allowed the chemicals to leach out of. So he was using this as a treatment, believing it's going to have no effect whatsoever on his patients. So interestingly, however, a number of patients seemed to get better with this treatment, and everyone was amazed. But the differences, or in terms of the treatment to the standard of care, didn't exist. The same number of people were responding. And he realized, knowing that this treatment had no effect whatsoever, this became known as a placebic remedy for rheumatism. And this is really where we get the idea of placebo effect, that no matter what you do in a study, you're always going to have individuals that differ in the treatment. I think a really important point here is we often see diseases where you see a relapsing, remitting nature. So you can have symptoms that are hard and really painful, and then they ease off. But some people, when they're taking a sugar pill, let's call it placebo, they may attribute it to that drug that they're having, and it isn't necessarily always the case. So we've seen here through history, we're starting to see some of the first signs of being able to pull together something that looks a bit more familiar now as to what we'd have holistically in a clinical study. We would be looking at, we need to look at different cohorts like James Lynn did. We need to have some comparison or placebo or other drug product to see, is there a real effect going on here? So these start to be the basis of what we would call modern clinical studies. But ethics, safety are not always a given. History teaches us a very hard lesson around the fact that not necessarily everything has been done for the greater good or for the good of the individuals involved. On the left-hand side, we've got a picture here of Nazi doctors. It looks like they're probably looking at someone who's been immersed in very cold water. Now, these are often prisoners of war. They didn't consent. they were just experimented on at the time. So the Nazi regime could work out better how to care for its own soldiers, in this case, its own pilots that could be coming down in the sea, and how do they potentially try and give them an opportunity to live longer in very cold water. So, you know, for many, you could say, well, this has probably done some good for pilots and the likes, but this was not taking an individual's primary life into any concern. In the middle here, we look at elixir of sulfanilamide. Now, back in the early 1900s, there was a bit of a craze here, and the sulfamides were very much a class of drugs that were often taken by people, a bit like antibacterials, but lots of people were taking them. But the Messengill company decided we're going to generate a new formulation here. So they thought about, well, what can we use as our base liquid that we add the sulfonamide into? And they decided to use diethylene glycol. Now, at the time, no one had ever tested it. It was a nice, sweet substance, but the reality is it's an antifreeze and it is highly toxic. So a number of people died as a result of taking this elixir that was supposedly there to treat them for their ailments. So here we see a case of, because no one actually looked at how safe a product was, it actually caused deaths. And then on the right-hand side, Tuskegee, I probably don't need to tell the audience in the US much about this, but obviously this was a clinical study performed at the highest level on black males that had syphilis, and they were telling them they were being treated and that they were going to follow them and make sure they had the best care possible. Now, those gentlemen did get access to some healthcare. They were regularly checked up. They regularly had their blood taken, but not a single one of them was treated for syphilis, even though penicillin was already available and could be used to treat any of them. These gentlemen were just told that they had bad blood. So again, I know that Bill Clinton made a very big apology for this, but again, just shows that even up till 1940s, 1950s, we were not necessarily seeing clinical studies being conducted in the way that they should. So I've talked about what's happened here, but how has that actually led to an evolution? So we've got a bit of a timeline here. And I think one of the key points to pick up here is post that sulfonamide poisoning, you saw the FDA, or Food, Drug and Cosmetic Act, launched the following year. So this is something at a US level that you had to prove that whatever your drug was, it was safe and effective. Obviously, then moving on through the 40s, after the Nazi war trial, then we see the Nuremberg Code brought out. So this is based upon all of those Nazi doctors who've been carrying out experiments. We saw countries signing up to the Nuremberg Code. And this was really talking about the first guidance on ethics for medical research, making sure people were consenting to this. As we move on through 64, 97, we see the Declaration of Helsinki. So this is really about ethical principles in medical research. And then in 1997, the International Council of Harmonization Guidelines. So they provide now a very big framework with which companies must adhere to deliver clinical studies. So I'm saying that at the moment we have safety nets in place. That's not to say things don't go wrong in clinical studies, but holistically, we see far better practices across the board. So what are these key principles? Now, this is a really busy slide. There's a lot on here, but there are 13 core principles of good clinical practice. Essentially, I'm not going to talk through all of these, but essentially at the heart of this is the safety, well-being of participants, and that their participants' rights are maintained. There's also another part that says we must keep your information confidential. Now, I understand in this technological age, we can do as much as possible to maintain that confidentiality, but we know that people get hacked. We know things can go wrong. So we have to maintain as best we can protection for all participants. The other thing that's really important is around the studies need to be ethical. They need to protect people, but at the same time, we need to ensure that we've got a body of evidence that gives us reason to believe that a new drug product would have a benefit to patients and that we should be testing it, and that we can do that in a high quality way with essentially medical decisions and qualified care teams being at the centre of that. So that is your treating physician at your hospital or wherever you're getting that treatment at the time. So those for me are really the key principles here, looking forward as to how to ensure that we undertake clinical studies that protect patients, but also deliver to be able to say we've got a safe and effective product. So we've talked a little bit about the history here. We've talked about some of the measures that are now in place to protect individuals in clinical studies. But how do we get to this point? What is the drug development process? How do we get a product into the clinic and into patients? So I think this is rather facetious, this slide, but we've got a new wonder drug. We give it to you and wonder what it will do. Now, to some extent, this is always going to be correct in that you can never predict what a drug will do in humans until you've tested it. Now, if we did know what a drug would do, we know that it have efficacy, we knew what any side effects are, we wouldn't need to do clinical studies. But the reality is you only learn about a drug through clinical studies within the human population. So what is the process of getting there? So on the far left-hand side, we've got discovery research. Now, this is an area where I spent 15 years of my life in pharmaceuticals, where you've got lab studies and animal studies. Now, the animal studies may be disappearing. I've obviously heard that the FDA are now looking at trying to get alternative approaches rather than using animal studies. But essentially, within your lab studies, someone is trying to think about a disease of interest and being able to say, what might be causing that disease? What pathway could we impact? And how do we make a drug, a molecule, an approach to be able to treat that? Then through that process, they look at developing a molecule, whether it's chemical, whether it's monoclonal, whether it's a cell therapy to be able to do that. They'll test it in vitro normally, so that means in a in a petri dish, so just with some cells, or maybe in an organ model of disease. Once they've believed that that drug works in the lab setting, they'll then probably move on to animal studies. Does it work in animals? Is it safe in animals? It's only at that point that you've got a full data package that suggests that you've got a drug that could impact the pathway, could give a clinical benefit, and is perceived to be safe, that you move into phase 1. So phase 1, you have a very small number of patients, and this is really where we start testing the safety of molecules. So we'll start at a very low dose and then start increasing dose levels just to make sure it is tolerated by individuals. Once we've completed that work, we'll move into phase 2. Phase 2 is very much then more participants, probably in the tens, sort of 40, 50, 60 plus. And then can we show the drug impacts those symptoms, the underlying disease, to have a beneficial effect? And also still looking at safety. Phase 3 is where we do the really big studies. So this is confirming within a patient population, within 2 separate phase 3 studies, that the drug says what we believe it will, and again, that it's safe. And then phase 4 is usually once you've got medicine approval, you'll continue surveillance there. And we see this with all drug products. Even once they're available to patients through prescription or over the counter, they have to continue collecting safety data beyond. So safety is always at the heart of everything that we do. Now, the problem we have in drug development or drug discovery as a whole is that we start off with some fantastic ideas. We've come up with more chemicals, monoclonal antibodies, than I think many would really be able to comprehend. But actually, what we see is as we start moving through the discovery process, some of those fall over because actually you can't turn it into a tablet, or it can't be turned into something that's inhaled. We'll lose some at the preclinical lead because they don't work in an animal, or we see some safety effects that we're not interested in. Then we see some drop out in phase 1 with safety findings. But as you'll see in this upside down triangle, we start with a lot of drugs and we end up with very few. So this is really labour intensive, and it is challenging to pharmaceutical companies as a whole to be able to really be able to predict accurately at the start what's going to be a great medicine at the end. So why do most of them fail? So the big bucket here on the left-hand side is they just don't give the patients the amount of effective treatment to really control their symptoms. 30% of them have unmanageable toxicity. Now, those normally get pulled very early, probably in animal studies or phase 1. Some of them have poor drug properties, so this will again be at the discovery end. And some of them are just poor strategy. They've gone into the wrong disease and they've failed because they thought they might have an impact there. So there are a few examples of that as well. But the one thing that we've got to remember, irrespective of the huge amount of drugs that start at the top and what really gets to the patients at the bottom, is that attrition that we see, those loss of drugs that we see through that process, is outweighed by the patient benefit of the few that come through. Now, I was thinking about having a slide here that just had drugs coming in left, right, and centre, showing you that there are thousands out there. But I thought it's really probably just worth highlighting a few of the big ones here. AZT, highly active antiretroviral therapies for HIV. This has been a game changer since the 1980s, where this really was seen as a pandemic and everyone was worried about it. These drugs now control viral load to such an extent you can't even detect virus. It's quite incredible. We've got insulin for diabetes, for those that have got autoimmune disease, that are losing, this key function in being able to control glucose. So again, fantastic. We've got statins now for heart disease. So we're looking at an aging population across the board, essentially because we have better medicines, because we have better healthcare. But it means we're all living old till an older age. So these statins are really helping that elderly population. Birth control pills as well. So this has been something that, from the 1960s onwards, it allowed females to really take control of their own bodies and decide and family plan when they wanted to. On the top right-hand side as well, we've got the anti-TNFs. So these have been enormous in autoimmune diseases. I could mention all sorts of diseases that anti-TNFs are now being used to treat. So again, these benefits outweigh that loss of molecules within the pharma funnel as we go through. I'm going to pause though here and try and convey a message that I think is really important. And irrespective of all the other medicines, vaccination is essential to individuals. It's essential to global health. Now, I know there are lots of stories that circulate around vaccination. Trust me, every country has them. Some believe that they've got microchips in them and they're trying to be controlled by governments. Others, here in the UK, we saw a massive scandal regarding the MMR vaccine when someone said, oh, this causes autism, you should buy my single shots rather than the triple shot MMR. We've seen cases in France as well. But the one thing I can say is that childhood vaccination as well as adult vaccination is so incredibly important. We have seen the eradication of smallpox, one of the biggest killers in the world, that was absolutely shocking for people to have. We've also seen polio be reduced to almost nothing. But vaccination only works in a herd scale. So we see cattle farmers using it. We see sheep farmers using it because they want to protect their entire herd. Now, we all need to vaccinate to make sure that we get protection, not only of ourselves, but our loved ones, our friends, family, but other people within our communities and areas. So this is my one key message. Irrespective of what you think about medicines, please vaccinate. This is so important to global health. So now I'll run into a little clinical studies overview. So I've kind of walked you through some of that already, but what are actually those drivers of clinical study design once we're actually here? So we've got safety, we've got efficacy, we've got dose. Safety, as I alluded to earlier, just runs throughout the whole process. And we see more and more people contributing to that safety as we go through the phases 1 through 3. So then we look at efficacy. So efficacy isn't really looked at in phase 1, but it can be. So dotted line there. But we tend to see it more in phase 2 and confirmation in phase 3. Dose is something, again, that we look to as we go through phase 1, at least show safety up to doses that we think will be efficacious and provide a benefit. But it's really in phase 2B where we do the full dose response. So, when we look at these and we think about safety testing, what is the quantity of drug that is tolerated without causing severe harm? Now, ideally, you just want that to be no harm, but don't believe that anyone ever tells you a drug is safe. Now, what we do know about all drugs is that they have a side effect. Whether these are described as adverse events, severe adverse events, those are those requiring hospitalization, whatever you put into your body as a drug will always have a side effect. So what we're looking for here is, can we balance that improvement in the patient versus what is an acceptable side effect? Now, that may be very different in myositis, where maybe, some pain discomfort isn't a good thing. But when you look at oncology and tumors, people will go through some very severe chemotherapy approaches there to get themselves into remission. So it's always balancing the weight of the disease versus what are the acceptable side effects. Now, proof of concept. So this is very much about saying, does the medicine, the drug, improve the disease? Optimal dose, ideally, what quantity of this drug works best in the disease without causing excessive harm or safety side effects? And then obviously the confirmatory over on the right hand side, this is very much about showing we've got a dose of drug that we know what it does, and we are happy to submit this in countries as a package to say we should be able to take this forward to patients to be able to get. So going back to these on an individual basis, safety. This is not negotiable. This is one where we have this at the heart of everything that we do. So based upon this allows us to select our patient population. So if you're conducting a study and you want to know how your drug works, you're not necessarily going to pick a patient population that you know may be at high risk of cancer or some other co-disease at that time. It also means that you are selecting the patient population that you think will get a benefit and be able to see those side effects that are acceptable to them. Dose and dosing regime. How much drug, how often do you give it? Again, this is all very much around the safety profile. How we monitor you. So within studies, you could have very frequent monitoring, you could have less frequent monitoring. More often than not, you at least have monthly safety monitoring when you visit site or call into the site. And then participant discontinuation and study stopping rules. So we need to clearly define, if a signal is observed, should a patient stop treatment there and then? Or is there a signal enough that enough patients are getting this, we just need to stop a study? So what we have to ensure is that when we conduct a clinical study, we are continually monitoring our patients throughout, and we are doing what is right and best for them. Efficacy. does this drug work? Does it have the benefit that you want? So again, you've got to select the right patients here. There's no point selecting patients, let's talk about myositis, where you want to see an improvement in muscle function if actually they're currently already in remission. They're saying that their muscle function has improved. Yes, they may still have the disease, but that's not the patient population you want to treat. You want to treat the ones who are not in that fortunate situation. Comparator. What are you comparing your drug to show that it works? Now, earlier, I was talking about the placebo effect. Now, this is one area where we can use placebo as a comparator. Normally, placebos are used in studies where there's limited options for the patients. That wouldn't be to say that you would remove them off other treatments. If they're on a standard of care, again, for myositis, if you're on steroid immunomodulators, you'd stay on them, but you may have placebo. In other studies, in, I don't know, rheumatology, you may look at, well, there's a lot of drugs already available. So you might want to use one of those already available drugs to use as your comparator, because actually no physician is really going to say to their patients, oh no, you should just stay on, this treatment and we'll test the drug. They will put you through your DMARDs and onto TNFs or other treatments moving forward. So that might want to be your competitor, another treatment. Medication standardization. So, can we standardize the way the drug is given to you? Is it a case of, well, we predict that we need to take this daily, weekly, monthly? So essentially what we don't want to do is give you a drug, you have an effect, and then you drop down and it's not having an effect the next day. What you essentially want to do is try and reach this plateau and maintain that plateau. And that's achieved by making sure you've got regular drug treatment that keeps you in that therapeutic window. And finally, is the change meaningful? And by that, we mean meaningful to patients. Is it having a benefit to you? Not a benefit that we think you should have, but a benefit that is of use to you. So I've already talked about the efficacy, keeping you up on the medication standardization, but how much drug? How do we define this? So essentially, we still choose the right patient population, but there are a number of different aspects within a patient population that can impact the dose response, as we call it. So how much drug to get how much response. We know that race and ethnicity has an impact. We know body weight can have an impact. We know lifestyle can have an impact here. So there are numerous examples here of where we can look at, even though we're a global population, there are enough subtle genetic differences here with race and ethnicity to see differences. We know that body weight increases, we see increased volume of blood, increased volume of body water, so you may need a higher dose. You may move to weight-based dosing. We know that lifestyle can have an effect. So you could be a smoker. Is that really going to impact anything that you're taking on board? We know that grapefruit juice can inhibit a cytokine that essentially means that if you're on maybe over-the-counter medicines for hay fever, it actually stops that drug being broken down and you have a greater impact of effect. which can cause increased drowsiness. So there are a number of factors to be borne in mind here when we're thinking about how much drug and what could be impacting that. So what does this mean for you as individuals looking forwards? So clinical studies at a glance, you've got consent at the start, and then we move through screening, dosing period, and safety follow-up. These are the main parts of a study. But the clear message here is no one is allowed to touch you in a clinical study, to take blood, carry out any procedures at all, unless you have consented. And that must be the first thing that is ever done before anyone even touches you for any of the procedures within the study. So when we look at screening, we might see physical examinations undertaken. So this could be height, weight, temperature, ECG to look at heart rate. So could be lung function. We'd have laboratory tests. So we may take blood, urine for those to be able to look at how you are at the start of the study to make sure we have got a baseline to assess your general health too. We'll also review medical history, but then you might also have some baseline assessments that are more defined to what is the end point in the study that we're really interested in. Do we need to know anything genetically about you or from an RNA basis to know whether this could impact you in the study? So you may look at, in oncology, BRCA associated with breast cancer. Are we looking at those sorts of things? And you may have to fill out a questionnaire, usually on an electronic device. As we move into dosing, it all looks very similar. It's the same sort of test because we're testing your health, your safety constantly, and we're assessing your response to treatment. The only difference is you're going to be dosed, whether that's an injection, an infusion, whether it's pills, whether it's a liquid, an inhaler. That is really the difference here. So what you do in screening, you'll probably continue doing throughout the dosing period. And then safety follow-up at the end of the study. We don't want to release you from the study until we know it is safe to do so, that we have followed up after the last dose of drug. You have had time for that to clear from your system and us be able to say anything untoward that could happen from now on is very unlikely related to the drug product. So with that, I'm going to close with a couple of final slides. And first and foremost is that it's only in partnership with patients that pharma are able to generate new medicines. And I think we have to continue to emphasize this. This is partnership. This is very much you're in control of your own clinical journeys if you wish to join a clinical study. You always have the option to opt out or opt in. So I think it's really important that we get that across, and that an individual may not benefit from a clinical study themselves, but it's only through those individuals that we now have treatments for the many. And that I think every patient who joins a clinical study are absolute heroes because they are really providing the opportunity for not only themselves but the next generation to benefit from treatment. I will leave with this final slide saying that my next presentation webinar will be based upon the Jasmine study, which is a clinical study that AstraZeneca are performing in polymyositis and dermatomyositis. And with that, I'll hand back to Paula and see if there's any questions that I'm willing to answer.
Thanks so much, Dr. Beaton, for that highly engaging walk through clinical trials, the lessons from history, good and bad, the global standards that we're all accountable to as we do this work together. As you said, it is a partnership. I learned quite a bit about drug development, and I know that our audience did too. We have many questions, probably many more than we'll be able to get to today. So we'll try, as always, to follow up with some attendees by email as best we can. And in the meantime, please keep the questions coming. We'll power through as many as we can. So we have several questions around the section of your presentation, what it means to be a participant in a clinical study, and a lot around eligibility and sorting through open trials. So let's just start with some basics. How would you advise that someone living with a form of myositis could find and volunteer for a trial?
That's a great question. And I think for me, having seen the Myositis Association website, I think that's a fantastic place to start, because I know you do list a number of studies from your corporate sponsors. So for me, that would be place number one. Go to your trusted resources, go to the people that you know that will be advertising these. For anyone else beyond that, you could probably do a web search, but the one thing that is used by industry is clinicaltrials.gov. So if you ever want to know about a clinical trial that's going on, you can always go into clinicaltrials.gov and search up a specific disease. Now, within that, when you find a clinical study, this may be answering another question. They normally list all of the sites who are participating in the study. So not only can you find out what the studies are, but they often list sites that are participating in the study. So you could identify from that where you may be able to go. I think this also brings up another important question in how much do we do to support wider engagement? Now, you can very easily just go to the sites who are conducting the study, but for me, it's really important that as a company, we speak to other surrounding sites, healthcare providers, to say, there is a study going on down the road. Please send your patients or make your patients know about this should they wish to get involved. So again, communication, partnership.
Absolutely. I will offer that the TMA website links out to the clinicaltrials.gov links for each trial that's active in myositis using the unique clinicaltrials.gov identifier, as well as where there are additional public-facing websites available. For example, AstraZeneca's site is autoimmunetrialandyou.com. So we link to both the industry-sponsored site as well as the US government agency site, and recommend that patients, work through the information. It's the same information, but it's interpreted in different ways and sometimes more visualized on the non-governmental website. So that can be very helpful as well. And thank you for mentioning encompassing more patients and families in clinical trials. We do have a couple of questions, including one from a TMA member who lives on Maui and does not often see trials coming to her immediate home area. So any advice for screening and enrolling in trials that do require travel, and in your experience generally, how would that work?
Yeah, that's always a challenge. But I think I've heard stories of people traveling enormous distances to be in studies, I must admit. So again, I think normally what you see within clinical studies is that often travel and the likes will be paid for, reimbursed. So there's always an opportunity. There's always an opportunity to speak to sites that may be involved and thinking about, does this travel fit with my own personal life? Because at the end of the day, some people do not want to be traveling huge distances and taking days over a visit. A, a clinic visit can take almost all day for some studies. So I think for those individuals, they need to think carefully about what they can commit to, how much they're willing to commit to it. Beyond that, do reach out to those sites or investigators that do have studies, reach out to the TMA, reach out to sponsors and say, how do I get involved? Because there are always ways for us to get patients to get to the sites and get treatment. So this is one where we take it seriously that we can't have a site in every town, city. You're limited by resource. So we've got to find ways to be able to support patients in achieving that. So don't be afraid to ask and make sure it's something you can live with.
Thank you. That feels like excellent counsel to me. And I'd like to assure the TMA members who are living with inclusion body myositis on today's webinar that while, for example, the AstraZeneca trial Jasmine is indicated in DM and PM, we are equally committed to advancing the science in all forms of myositis at TMA. And we do everything we can to help advance science. So there are a lot of ways that you can participate in things like patient experience focus groups, panels, advisory boards, and the like. And all of that helps because it helps TMA better inform companies like AstraZeneca and the many others working in autoimmune disease about the key concerns of the myositis community. So it does, as you said, it is a partnership. Now, we also have a number of patients in the audience today who are living with polymyositis, and we have an excellent question from one of those attendees who says that the label of polymyositis is already outdated, and many patients like me are now reclassified to IMNM or IBM. What is the process to handle future updates on myositis classifications and accessibility for drugs if they come to market for PM?
Yes, now this is an interesting one, and we're obviously seeing a shift in the nomenclature, and I think we aligned with some nomenclature or naming strategy that was very much said, well, polymyositis includes the antisynthetase, the overlap, the IBM, the IMNM. So we know that IBM and IMNM do differ in their underlying cause. So when I talk about polymyositis within this clinical study, it's essentially antisynthetase and overlap. I agree, the language is a little dated, and we are working with experts in the field to sort of think about how do we better name these and make sure that that is something that moves through the field. So these things, unfortunately, just have a bit of lag time. I do wish, though, Paula, to maybe speak to one of the questions that I've seen
Sure.
regarding why are exclusion criteria so strict?
Good question.
So
Important question.
it is, and it's essentially the heart of my presentation as to how do we show a drug works while showing it's safe and picking the dose. So essentially, within any clinical study, you want to understand ideally the truth. What is the truth? The truth is, if you treated everyone in the world with that treatment, you would know the truth, how it works in every single person with it. But the problem is that isn't feasible. So what we have to do is try and sort of project what we believe that truth is going to be by selecting a patient population that we think will show a benefit. You're essentially trying to reduce the number of variables in your clinical study so you can say within this population it is safe, it works, and we know how much drug to give you. So with that, you often see, so let's talk about myositis itself. You want people who have got enough muscle or impact on their muscle function to demonstrate you can improve it, but you don't necessarily want the muscle function so bad that actually they're never going to show an improvement. So you've already taken this whole and said, well, actually, what can we achieve within a 1 year period? Now,
Mm-hmm.
for those that have such little change in muscle function, you can't show a difference very easily. You'd have to have an enormous study to show a tiny difference. So with the numbers that we're talking about, we have to define enough of a difference in muscle function to say we have shown a meaningful change. So that speaks to one aspect of the disease characteristics. Then beyond that, you may think about, well, actually, we want to discount people who have maybe had recent surgery or cancer, because whenever you're doing a study, every event that happens has to be reported. And I'm aware of clinical studies that just have been unlucky that a couple of people have ended up on the study and have maybe they've had cancer. But because of the way we randomize, we don't always, you could end up with both of those on drug treatment and not in placebo. Suddenly everyone goes, hang on, your drug causes cancer. Actually, it doesn't, but that has been the impact of allowing people in who are maybe at a high risk of cancer. So what we see is, as I said at the beginning, we see a contraction of the eligibility criteria to say, are we safe? Are we effective in the population? And then can that be extrapolated to the wider population? So this is a reason why we see a lot of people who do want to enter a clinical study are unable to do so, because we have to control the study as much as possible to show those effects in terms of safety, dose, does it work?
Thank you, Andy. And I just want to echo that while it can be frustrating if you're screened for a trial and not enrolled, or if you're enrolled in a trial that's discontinued, again, the nature of the partnership in drug development means that there are some very hardworking, big-hearted expert people like you, Andy, who are similarly frustrated, right? Because you work on many molecules that are not able to surpass those hurdles and help patients. So, one interesting question, if I can ask you just two more questions, we are running a little bit over, but I don't want to miss the
That's fine.
opportunity to ask these very thought-provoking questions. One attendee asked, how can patients help to push for quicker regulatory approvals of drugs? And, that's a big question, but it does tie to TMA's work in awareness and advocacy. And I'd love to put you on the spot and get a few words of wisdom from you if we can.
How do we get to faster approvals? That's a very good question. I mean, do you mean in terms of once we submit, or are you just talking about the drug process as a whole to getting to approval?
The audience member said regulatory approval, so I'm wondering if that's point of submission, for FDA decision
Oh,
making.
sorry. Yeah. So Yeah, this is an interesting one because there is a defined period from submission date to getting approval, and that is very highly defined by whether it's the FDA or the EMA. We are seeing within pharma industry now, everyone is trying to contract that timeline from study finish to study submission because actually they don't own the approval timelines. Those are owned by the FDA, the EMA, other health authorities. I think all we can do, patients, pharma, can only really try and lobby the health authorities and look to expedite those timelines. Now, we know for very rare diseases, they can be shortened because there is such a very high unmet need in some of those awful rare diseases. But essentially, we're constrained by health authorities within that timeline.
Thank you very much. And final question, another big one. Any thoughts on how AI will help in drug development or help to ensure better patient safety in the future?
Yeah, great.
I
Now
saved the
this
hardest
is
question for last. You're welcome.
well, it depends. Are we talking organic? Are we talking? So, yeah, AI. You can't get away from it. It's everywhere. Obviously, all of the companies at the moment are using AI to accelerate drug discovery, the early discovery. They're using it within sort of I mean, you've got to be very careful in clinical studies because once you once you use AI, if you're using it for an endpoint, it has to be a regulatory approved. But that doesn't mean you can't use AI tools to help support safety monitoring or the like. So I think what we're going to see is a lot more of that sort of work going on where it's used as a tool, but it always has to be checked by a real person. Do I believe AI is good enough at the moment to take over the job of my study physician or my clinical scientist? No, but I believe it's a tool that they can use to simplify their work and free up more time to do, you know, really important aspects of looking into those patients that maybe we are worried about or looking at more closely. What I can say is that I have worked on using machine learning as well for clinical endpoints, and that I think you're going to see a very big change in the near future, whereby, especially with imaging technologies, we're going to see where you have this intra-observer or inter-observer variability, where one one doctor may go, oh, that looks like a score of 2 to me on that ILD scan. And everyone goes, oh, it looks like a score of 1 to me. We know that that difference occurs everywhere, and that's based upon your own perception of the image. But we are seeing more and more machine learning approaches being used to view those images and be able to score them more accurately. So as I say, at the moment, I'm not necessarily aware of any being specifically getting regulatory approval as an endpoint, but we are starting to see that. And in fact, I've launched an artificial intelligence scanning machine at a hospital to see how it works and to make sure that actually it simplifies the physician's life moving forwards. And they can use it to train the trainee doctors coming through as to how to score more effectively. So it could cause standardization. So all of these are very positive aspects.
Thank you so much. That's fascinating. And I really appreciate your depth of knowledge and also the depth of tolerance for the questions about the past, present, and future of clinical trials. So thank you again to our expert speaker, Dr. Andrew Beaton, and thank you again to AstraZeneca, the sponsor of today's webinar. And for those of you who joined us for the first time, TMA, or the Myositis Association, envisions a world without myositis. And every day we're working toward that goal together with all of you. So we appreciate your participation and engagement today and look forward to seeing you on our next webinar, which is an Ask the Expert program with TMA Medical Advisory Board member and neurologist, Dr. Elie Nadoff. That's happening on October 23rd at noon Eastern. So same bat time at about 8 days from now. Look forward to seeing you. And Erica has kindly put the link in the chat. So thanks so much to Erica, Linda, Rachel, Jackie, and the TMA team. And thanks again, Andy. And we'll see you soon. Stay tuned for the next webinar in this series.
Thank you very much. It's an absolute pleasure.